This is our final lecture of the semester. In this one, we're going to be looking at chapter 16, light and vision. We're going to look at the eye. And although it's just vision, there's a lot of things that have to go into how we perceive photons of light into color, how do we perceive color into objects, and how do objects start to hold meaning for us in our visual cortex. So there's a lot of things that we're going to do for this. So it will be a little complicated, but remember, pay attention to the items that are in red. Those are the ones that are going to be directly asked on the test, and they're the ones that you can find on your study guide. So vision, perception of objects in the environment. And this is by light that they either emit, like a light bulb, or reflect pretty much everything else. A chair, your clothes, everything else is going to be reflecting light. And light is visible electromagnetic radiation. Our vision is limited to the wavelength of 400 to 750 nanometers. And nanometers is a very small measurement. So we're only able to see 400 to 750 nanometers. And if you remember back when you were in elementary school and you first drew a rainbow, or you first saw a rainbow, you saw red up on top and you saw purple in the middle, or in the bottom. So ultraviolet is going to be below 400. And you might not have ever wondered before, why is purple on the bottom of a rainbow? And it's on the bottom of a rainbow because it has the shortest wavelength, so it bends the most. Red, on the other hand, has the highest wavelength, so it bends the least. That's why when you see a prism that is going to reflect visible light, red's on one side with the highest wavelength, purple's on the other with the lowest wavelength. So ultraviolet, which is below violet, is going to be below our ability to see violet or purple, and that's below 400. Infrared, above red, is going to be above the 750 that we can see. So we might not even know that it's there, but ultraviolet has a lot of energy because they're very small wavelengths. So they're very, very small, which means small, they have a lot of energy. They can destroy macromolecules. Infrared, they have very long wavelengths, so they don't really do much. They might cause some warming of the tissues. If you have an infrared gun or a little light, it might warm your tissues to muscle relax and stuff like that. So all the other wavelengths we can't see. Some animals see different wavelengths. So just because your dog sees you, they perceive you a completely different way. So our cells only have the ability to see between those two wavelengths. And light must cause a photochemical reaction to produce a nerve signal, which is why we can only see between 400 and 750 nanometers. We do that by using all the different structures of the eye. First one, conjunctiva. And the conjunctiva is that outside layer. You might have even heard conjunctivitis, which is going to be the infection within this layer. It's caused by a bacteria or viral infection. Sometimes you might get some debris or something caught in there, which can cause a response. It's highly vascular, which means that it has a lot of innervation in blood vessels, which means it can heal quickly. But also, if you have some bacterial or viral infection or debris, those blood vessels are also going to cause inflammation and redness. So if there's redness, people call it pink eye, that's just conjunctivitis or infection or inflammation within the conjunctiva. You might not have ever even thought about this either, but tears. Tears are the way our body can flush the eye and keep bacteria, can keep viruses and other types of infections from infecting the eye. Because this is going to be saline-ish, it's going to be able to keep it at a certain pH and a certain environment to be able to provide nutrients to the conjunctiva by oxygen and nutrients, Also, that saline is going to wash foreign particles and other bacterial or other types of viruses, infection in the eye, which then drains from outside to in or lateral to medial into these ducts and eventually into the lacrimal sac and into the back of the nasal cavity. So if you've ever wondered, what do your tears taste like? You all know they taste like salt. Why can we taste salt? If you think about that, think back to your taste, to think back to micro lecture number 1. Where do we have salt and where do we have that salty taste buds? It's going to be in the back of your tongue. So that's why you can taste salty tears, because them flowing down that sac down to the back of your pharynx. What are the nerves that innervate these muscles? So we have 6 muscles. 2 that run at an angle, 1 on top, 1 on the bottom. They're called inferior oblique, superior oblique. Then you have 4 muscles that run straight back that are called rectus. Superior rectus, lateral rectus, inferior rectus, medial rectus. Medial rectus is next to the nasal bone. Lateral rectus is zygomatic and frontal bone up here. So outside of the skull, inside of the skull. And an easy way to think about it is if you have the entire eyeball, you can see the lacrimal duct. Sorry, the lacrimal gland is going to be lateral. The lacrimal duct is going to be medial. Another way to look at it, the superior oblique muscle is going to be going around this little hoop right here called the trochlea. And you might not think the trochlea is that important, but look what the name of the nerve that innervates the superior oblique muscle. It's called the trochlear nerve. And if you activate or constrict this superior oblique muscle, your eyeball goes up and in. If you innervate the medial rectus, it turns your eyes medially. Lateral rectus turns it lateral, superior up, inferior down. And then the inferior oblique muscle turns the eyeball down and away. So abducens abducts the eye away from the midline. Ocular motor is going to do all the other eye movements. So kind of knowing which eye helps you move up. Superior and inferior oblique muscles turn the 12 o'clock pole of each eye toward or away from the nose. All that means is this one goes towards the nose, this one goes away from the nose. Don't kind of think about that. And then which cranial nerve moves the eye in certain directions. And this is pretty much the main area of the eye. This has everything you need to know. So everything we're going to talk about, pretty much, except for the rods and cones and the retina, is going to be in this one slide. So I'll go over all of these things in a lot of detail, and then I'll just kind of repeat myself on a couple of the slides that follow. But it can be broken down into optical or non-optical. And you're like, optical? Light passes through it. So anything that light passes through, all the way back to the retina, is called an optical component. So the cornea. Cornea is the outside layer. Behind the cornea is the anterior chamber before you get to the iris. Iris is a not optical component, but the space that the iris makes is the pupil. The pupil can get bigger or smaller. That's going to be dilation or constriction. If it becomes bigger, more light can come into the retina. If it becomes smaller, less light comes in. Less light would be when you're in a very bright situation. When it's larger, more light can come in, so it'll be in a lower light situation. Think about that time when you're in the movie theaters and you walk out on a sunny day and you almost get blinded. It's because your iris is very big, because a movie theater is usually dark. When you walk into the parking lot with a lot of sunlight, it hasn't constricted enough for the difference, so you almost get blinded for those first few seconds. Right behind the pupil is going to be the lens. The lens is what helps you focus light. So if you're looking at this or you're listening to it and driving, you can actually look at your steering wheel, look far away, look at your gauges, look at the cars, or if you're watching TV, you can look at your TV, which is far away, to your phone or iPad, and you can see how quickly it focuses the light. Because this is all connected via the brain, what happens is when you're about to look away, these suspensory ligaments and ciliary bodies are starting to stretch and to refocus where it thinks the light needs to be hitting the retina. So if it's far away, it needs to stretch. If it's close, it needs to relax. And we'll talk about this relaxing, getting wider, and stretching thinner, and how it focuses light later. But the lens is held in by these little suspensory ligaments or strings that go into these ciliary bodies that constrict and either make the lens fat or thin. Then behind all this is your vitreous humor within the vitreous body. The vitreous humor is very interesting. If you're in the lab class and you get to dissect in the in-person lab class and you get to dissect the cow eye or the sheep eye, when you cut it, all this gelatinous goo comes out. All of that's the vitreous humor. And it's important to kind of know that the vitreous humor is just there. It has to be completely clear because it can't block any of the light coming in, but it has to be kind of jelly-like so the eye can hold its shape in this certain shape. And then after the vitreous humor, you have the three tunics. You have the retina, choroid, and sclera. And in the retina, you have an area called the macula lutea and the fovea centralis. The macula lutea is like your dartboard in the back of the eye, where you have a lot of cones. A lot of cones, high definition vision. In the center of that dartboard is the bullseye. That's the fovea centralis, where you want to look if you want to focus on something. If you look straight ahead, whatever is right in front of your vision is all those photons of light are hitting your fovea centralis. If you look off to the side, or if you hold your hand to the side of your head, that's blurry. Those lights, or light from your hand, is hitting this part of the retina. That has cones, that has some cones, but probably more rods. So it's blurry. You can kind of pick up definition. This is where your peripheral vision is hitting in your eye. Then all of these neurons from the retina are going to be collected by ganglion cells we'll talk about. They make axons that make up the optic nerve. The optic nerve exits the eye via the optic disc or blind spot, and then eventually those axons form the optic nerve. That's it. We talked about the optical component that light passes through. We talked about the neural component from the retina and the optic nerve. If you listen to this a couple times, you can pretty much skip the next 20-something slides. So, optical component for more information, transparent elements that emit light rays. They can bend them, they can focus them, but pretty much what they're doing is they're just trying to focus on whatever you're doing, whatever photons of light you're looking at. They're trying to focus them on the retina. The cornea we talked about, the outer surface, the aqueous humor is between the cornea and the iris. The aqueous humor is very important. We'll talk about that with fluid buildup in the eye and what's that called. Here is how the ciliary bodies kind of keep processing the anterior humor within the anterior chamber, and it's released by the ciliary body into the posterior chamber, passes through the pupil to the anterior chamber, and then it's reabsorbed into the canal of Schlemm right up here. And then this is how this continues to be reabsorbed, cleaned and reabsorbed. The lens is lens fibers, flattened, tightly compressed, transparent cells. They're held together by suspensory ligaments, and then those suspensory ligaments go into the ciliary bodies. You have the vitreous body, which holds the vitreous humor. That's that jelly-like structure that fills the space between the lens and the retina, which makes the lens, or makes the eye hold its shape. When you look at the neural components, you usually see a picture like this. Right back here, that's your optic nerve. Over here, that's your macula lutea, and in the middle is your fovea centralis. So this area right here, you're almost looking through the retina. And most of those blood vessels are right behind the retina. And blood vessels and everything else are coming out with the optic nerve. That's what you're looking at when you see that. If you see blood pooling over here, that can be wet AMD. You can see a lot of different things from this type of view of the retina. So if you go to the doctor, the eye doctor, and they look at you with light, that's what they're looking at. They're looking at this picture of your retina. So neural components, retina and optic nerve, I kind of mentioned that. Retina is going to have a bunch of different layers. We'll talk about how the rods and the cones are going to interpret photons of light differently, and then how the ganglion cells are going to be the output neurons of the retina and how they make the optic nerve later on in this lecture. We also looked at the ophthalmoscope, which that picture, we looked at the macula lutea and the fovea centralis, and then the blood vessels of the retina coming through with the optic nerve. This is just it again, where you can see the fovea centralis, the macula lutea, the optic nerve, and the blood vessels. You also have a blind spot. I mentioned that the optic disc was also called the blind spot, and that's because there's no rods and cones in that part of the retina. All you have is axons leaving the retina. So because of that, if you printed this out on a card, or you could do this too, where you cover one eye and you just kind of go in and out, what happens is the red dot or the X falls on where your optic disc is, and then it will disappear. So if you're staring at the X and move your head in and out, the red dot will eventually disappear. Now, if you're looking at a big screen, you have to go far back. So that's why usually it works if you take a 3x5 note card, you print that out, and then you can kind of move it in and out. You can see it. And what you see is you don't see the red dot anymore. You just see the green bar going across, and then the red dot disappears. If you are looking at the red dot and then moving that, then this X will disappear and the green dot will go through that. But again, if you're looking at a big screen, you're going to think I'm lying to you just because it doesn't work, because you have to go further back. And cataracts are going to be the clouding of the lens. So these lens fibers will start to darken, and if anything interacts with the ability for light to pass through, this will stop light from coming through, and what happens is they get replaced with natural lenses, with plastic ones, so you can still see clearly. If it's in the lens, it's called a cataract. A glaucoma is when the pressure is elevated in the eye due to obstruction of the sclera venous sinus and improper drainage of the aqueous humor. So that aqueous humor kind of passes between the pupil. It either gets blocked and the pressure builds up, or there's too much of the aqueous humor being pushed in. So it can be the drainage, or it can be some type of obstruction with the venous supply. So it can cause death of the retinal cells due to compression of the blood vessels. The blood vessels don't allow oxygen to go through. It can also cause a halo. It can cause flashes of lights just because of this pressure. You also have either refraction. So what happens here, you have light that's going to pass through different mediums. If you put something in front of your eye, like a lens for glasses, it's going to bend the light a certain way. That's what refraction's doing. So if you're looking at something through water, looking through something through air, that's why if you're looking at a fish from a boat and you try to shoot a fish, you're never going to hit it because the light is bending at a certain refraction and you're aiming at the wrong area where that fish is. So light passing through the center of the cornea is not bent. So it's coming through, coming through, coming through, not bent. Then it goes through this aqueous humor, bends it. Goes through the lens, which is a different structure, bends it even more, so it hits exactly on the retina. If you're seeing something very sharp and clear, it's hitting exactly where you want it to on those rods and cone. If it's blurry, you can either refocus or wear corrected vision, like you can wear contacts or glasses, and that will cause it to hit exactly on the retina where you want it to come. Something here called convergence of eyes. So near response, adjustment to close range vision requires three processes. Convergence of the eye, moving them towards the object, which muscles are involved. If you're bending the eyes in, it has to be the medial rectus. You can have constriction of the pupil, so you're actually going to let light in to try to focus on something. You're then going to have the accommodation of the lens, which is going to change the curvature that allows you to focus on nearby objects. And what happens here, far away, you're now reading a book that's close, you have the medial rectus that starts to bend, have the eyes start to focus close. You're going to have the lens start to change its shape that allows you to focus on stuff close up. If you're looking at stuff far away, the muscles have to move, the lens has to change shape, and then you can look at far away. Some people can see far away, some people can see better close up. The reasoning is the muscles and the lens. Maybe the lens doesn't stretch far enough for you to be able to see close or far away. So let's take a look. So common defects. So if you have perfect normal vision, light comes in that you're looking at and it focuses directly on the retina. What happens if the focal plane becomes right behind the retina? That's farsightedness. You do that by having a convex lens in front of your eye, either contact or reading glasses, that just allows those rays of light to bend more to bring it further ahead. If the focal plane is right in front, which is nearsightedness, you need a lens that bends them out a little bit, and then your natural cornea and humors and the lens can then refocus it back to where it's supposed to be. So myopia is nearsightedness, hyperopia is farsightedness. So how do we get these signals? So conversion of light energy into action potential, it all occurs in the retina. The structure of the retina, the pigmented epithelium, is the most posterior part of the retina. The pigmented epithelium in some books are right behind the retina. So when I show you the picture, I'll show you why sometimes that's considered part of the choroid. But what the pigmented epithelium do, they absorb stray light. So there's no blurriness. There's no type of other, those extra photons. They don't excite, they don't try to interfere with other rods and cones. What they do is they get absorbed by this pigmented epithelium, so they only are focusing on those rods and cones that you want to be activated. And what happens here is you have photoreceptor cells, and these are going to be rods and cones. What they do is they absorb light and generate a chemical or electrical signal. It says certain ganglion cells. We're going to skip those for the time being. Only rods and cones produce visual images. And what happens here, rods are going to be a certain wavelength of light. Cones will be a certain wavelength of light. We'll learn about that in a little bit. After the rods and cones, you have horizontal cells. And then after the horizontal cells, you have bipolar cells. These bipolar cells, synapses with rods and cones, and then pass things back. Eventually, they reach the output neurons called the ganglion cells. These are the largest, and their axons, the signals that they're sending away, are going to start to form the the optic nerve, cranial nerve 2. So what happens here? A rod, the tip of the outer segments looks like a rod. The cone points to a cone. That's why it was called a rod and a cone. Ramon Cajal and a couple other scientists around end of the 1800s, they used different types of silver nitrate to stain things, and they're like, ooh, look at this in the retina. There's something that has a rod shape and something that has a cone shape. So that's why they're called rods and cones, because underneath a microscope, that's what they look like. So rod cells are mostly going to be in night or scotopic vision. Monochromatic, usually going to be purple to dark. So if you kind of see a blue hue at night, you're kind of picking up all the signals from rod cones. And it's low light because blue-purple is very low wavelength. That's why it kind of has that type of look. You have a cone cell. You have 3 main types of cone cells we'll talk about. Those are going to be color or photopic vision or day vision. Also, a term that's kind of meant to this or attached to this is high definition vision. So you see a lot more detail with cones than you do with rods. Think about how you see during the day versus how you see at night. And this is what we look at. So this is the choroid, sclera, the back of the eye. All of this is going to be in the retina. This is the front of the eye. So unlike most of the systems in the body, it's kind of situated backwards because light has to pass all the way through the ganglion cells, this layer that has amacrine cells, bipolar cells, horizontal cells. These are the nuclei of the rods and cones, and these are the outer segments of the rods and cones. So light has to pass through all of this before it starts sending the signal back through the light and out the nerve fibers to the optic nerve. You can see why the pigmented epithelium is right here, and those stray photons of light just get reabsorbed into that pigment. And then right behind that's going to be your choroid with blood vessels and everything else. And ganglion cells are going to be the end and the output neurons of the retina. And this is kind of what it looks like, where you see the back of the eye, you see how the pigmented epithelium is surrounding them and just absorbing those stray photons. One strange thing about this, we're not really going to get into a lot of neurotransmitters, but glutamate release is the main one here. And what happens here, you have the horizontal cells that kind of integrate rods and cones together. You have bipolar cells that can do a rod or a cone or can do just specific cones. And this is how you can do different types of vision using the same types of photoreceptors. You might see colors differently. It's all because of how the signals taken in by the bipolar cells. Bipolar cells, there's amacrine cells in this layer right here that then integrate the signal and modify the signal from the bipolar cells before it reaches the output neurons, which are the ganglion cells, then off to the optic nerve. And again, the direction of light comes in, the signal goes the opposite way. So we talked about horizontal and amacrine cells just in passing. This says they do not form layers within the retina, but if you asked any retinal scientist, they definitely do. So and that's why I didn't really get into that. Horizontal and amacrine cells form horizontal connections between cones, rods, and bipolar cells. It enhance perception of contrast, edges of objects, moving objects, and then not just moving objects, but what direction is that movement going, and changes in light intensity. And it's those combinations of how they all make those horizontal connections and how all these things work. And we're not getting into any of these because just like me, you can get a PhD in just vision. And I got my PhD in bipolar cells. So there are 19 different types of bipolar cells, or 23, or 30 something, depending on what model you're using, as in like mice and rats or human. So much of the mass of the retina is in the astrocytes and other glial cells. All of these types of astrocytes or glial cells are just helpers. They're just trying to assist with either the blood flow, trying to keep it out, also trying to make sure that the retina itself, and not just the retina, but the inside of the eye with the vitreous body, is away from the immune system of the body. So it has its own type of way it deals with infection. A lot of gene therapy happens in the retina because if you inject something into the retina, the immune system cannot fight it off because of those astrocytes keep all those parts of the immune system outside of the eye. So there's a lot of different things and a lot of things to learn just about the eye. And we're all skipping those because there's way too many things. Rods contain the visual pigment rhodopsin. So if you hear rhodopsin, it's going to be part of the rod. And it's going to be visual purple. So that's why in low light, you see that bluish kind of low light type of thing. There's two major parts of the molecule. There's the opsin, which is the protein portion embedded in the disc membrane of the outer segment, and the retinol or retinin, the vitamin A derivative, and it has an absorption peak at the wavelength of 400 nanometers. It cannot distinguish one color from another. If you remember, our lower end of the vision was around 400, 450. So this is at the lowest end of our vision and our wavelengths. And it's all because this one protein only spikes or only becomes activated or changes when this photon of light or this wavelength of light comes in. Cones, on the other hand, contain photoopsin. It's very similar to rods. The opsin contains different amino acid sequences. Because it has different amino acid sequences, it can determine wavelengths of the lights absorbed. So that's why we can say there's 3 different types of cones. They look exactly the same underneath the microscope. So you have no idea which ones they are. But if you looked at the opsin molecule and you looked at the amino acids, those amino acids can tell you which wavelength of lights they're going to absorb and become changed. So this is kind of generating the optic nerve signal with light and dark. So in the dark, I always start kind of over here. We're just going to kind of reabsorb and kind of add up these different types of molecules. So the opsin and cis-retinol enzymatically combine to regenerate rhodopsin. So opsin and retinol, they become combined. Light comes on. Rhodopsin absorbs a photon of light. This changes. It's bent, no longer bent. Because it's no longer bent, opsin triggers a reaction cascade that breaks down cyclic GMP. This stops or cessation of dark current, signals created in the optic nerve. Then the transretinol separates from the opsin, then goes back to transretinol is then enzymatically converted back to cis-retinol, and then it gets put back into this molecule. So right when photons of lights hit it, it changes, and it can then signal the optic nerve to create a signal. This is confusing. The reason why it's confusing is on the next slide. Before I get there, I want to draw your attention to the retinal disassociates from option or bleaching. After a while, if you're looking at light for too much, you will stop being able to see a certain light. That's called bleaching. So you don't really see this. Eventually, you'll just close your eyes, because if you're looking at the sun, yes, you're going to be damaging the retina, but what's happening is all these photons of light are changing cis-retinol to trans-retinol, and eventually you're not going to be able to transmit a signal to the optic nerve because you can't reabsorb and change it quick enough. Cones are much faster to regenerate their photoopsin, so 90 seconds for 50%, which means that's why the cones are going to be available when it's light outside and we see color. And cones we see pretty good in the beginning, but the longer you're looking, the less likely you are to see different colors. Try it out tonight and see if when you're walking around in the dark, you first open your eyes up in the middle of the night, you can kind of see around you, but then eventually that gets a little bit darker. It's not because you're going to a different room with less light or moonlight. It's just because this cis-retinol can't be changed quick enough from cis to trans. Now, another thing that's really strange. So generating visual signals. When there's no light, the rod in the cone is going to release glutamate. Glutamate inhibits the bipolar cell and horizontal cells. In light, that cis to trans stops glutamate transmission. And because it stops it, it allows the bipolar cells, horizontal cells, and amacrine cells to then start releasing onto the ganglion cell. So this is very strange. On every other type of neuron and every other type of cell we talked about, when the brain wanted to produce an action, it would release a neurotransmitter. But in this case, when it's light, it's actually inhibiting the neurotransmitter, and then inhibiting or stopping an inhibitory neurotransmitter allows the other cells downstream to become active. It's not in red because it's very confusing and too many students get confused by it, but it is something when you are studying the eye. This is another reason why it's confusing and difficult to study. So in the dark, rods generally steadily release neurotransmitter glutamate from the distal end, from the basal end of the cell. When the rod absorbs light, glutamate stops. Bipolar cells are sensitive to these on and off pulses of glutamate secretions. Some bipolar cells inhibited by glutamate, others get excited when cessation stops. In this turned, inhibited, or excited, these cells are excited by raising light intensities, and this is how you can tell if it's really bright or if it's really dark because of this type of flexibility. I told you there's around 19 different types of bipolar cells in the mouse that I studied. So all of these different types of bipolar cells is how we can start to interpret light intensity and all this other stuff. Other bipolar cells are excited by glutamate and respond when the light intensity drops. So all these different types of cells are how we're actually doing this interpretation of these photons of light. So bipolar cells detect fluctuation of light intensity. They stimulate ganglion cells directly or indirectly because if they're either turned on or turned off. The ganglion cells are the only retinal cells that produce an action potential. Every other cell up to the ganglion cell is either going to release a neurotransmitter or inhibit a neurotransmitter. And ganglion cells respond to the bipolar cells with rising and falling firing frequencies. Via the optic nerve, these changes provide visual signals to the brain. So in the light, walking out into sunlight, the pupil constricts, closes up, and pain from overstimulated retinas. That's when you walk out from a movie theater into a very bright parking lot. The pupil constricts to reduce pain and intensity, Color vision and acuity below normal for about 5 to 10 minutes, and we've all been there before, so you know that this actually happens. There's that 5 to 10 minutes is needed because the pigment bleaching to adjust the retinal sensitivity to the high light. And rod vision is not functional because they're completely bleached. That cis to trans can't be regenerated in time, so you can't see any rod vision during this time. Dark adaptation. Dilation of the pupil occurs. Rod pigment was bleached by the lights. In dark, the rhodopsin generates faster than it bleaches. In 1 to 2 minute light or scotopic vision begins to function. After 20 to 30 minutes, the amount of regeneration of rhodopsin is sufficient for eyes to reach maximum sensitivity. Try that. Turn off the lights. Open your eyes in about a minute. See what your room looks like in the dark. Close your eyes for about 30 minutes. Open your eyes and see what your room looks like in the dark. After 30 minutes, it looks or seems brighter than it did after just 1 minute. And it's because of that regenerated rhodopsin. So dual vision. This is another reason why it's very confusing. So duplicity theory of vision explains why we have both rods and cones. A single type of receptor cannot produce both high sensitivity and high resolution. It takes one type of cell and neural circuit for sensitive night vision. It takes a different cell type and neuronal circuit for high resolution or scotopic daytime vision. So let's look. We have a bunch of rods, bipolar cells, generally attached to many of them. They were all synapsed with the ganglion cell. They go through scotopic vision to the optic nerve. Photopic, which is color or high definition vision. Usually, the bipolar cells attach one cone to one ganglion cell. And then all of those will start to go to different optic nerves. This isn't exactly what happens, but generally this is the pathway, because this one right here is a specialized cone cell. This one's a specialized cone cell. Where they are, the color needs to be different. If you look at a pixels of your TV, if you have an older TV, you can probably see that. That's kind of the way these are working. And because these are all close together, our vision kind of maxes out around 4K. Anything higher than 4K, you might think you can interpret it, but unless you have a very large screen, you won't really be able to see a difference in 4K and 8K because you only have so many rods and cones. So physiological evidence is going to tell you that you can't really interpret the difference between those two different types of TVs unless they're bigger, which is why most 8K TVs are 72 and higher, or longer in length. So rod sensitivities reacts even in dim light. They have extensive neuronal coverage, which means they have a lot of bipolar cells that attach to many of them. 600 rods converge on one bipolar cell in some cases. Many of the bipolar cells converge on ganglion cells, and this means that it's blurry. We don't see very high definition with rods because each ganglion cell is showing us inputs from over 600 rods or thousands of rods. The edges of the retina have wider spaced rod cells that act as motion detectors. That's why your peripheral vision, you can see movement. You can see stuff like that, but you cannot see finely detailed images with your peripheral vision. Your fovea centralis contains only 4,000 tiny cone cells, no rods. So your fovea centralis, or your fovea for short, has the highest concentration of cones. Each foveal cone cell contains a private line to the brain. So one pathway from a cone cell to a ganglion cell. You cannot have enough space in the eye for your fovea to be very large, because think about that. If you had your fovea was twice as large, the surface area of that would be over 4 times as large, which means the axons from all of those ganglion cells will start to block the photons of light coming in. And then even though you have a large fovea, you won't be able to perceive better vision. And that's another reason why vision is sometimes the most complicated thing in the body. And high resolution color vision, little spatial summation, less sensitive to dim light because it needs high energy photons to activate these cone cells. So primates have well-developed color visions. Nocturnal vertebrates sometimes only have rods. Nocturnal because they feed at night, so they have lower wavelengths of light to deal with. 3 types of cones are named for their absorption peaks. And these are the absorption peaks by these amino acids in the photoopsins. So short wavelengths have around 420, medium wavelengths around 531, long wavelength or high is around 558. And you might say, okay, so that's why sometimes you might say you have a blue cone, a green, and a red, but it's really, you can see the absorption peaks. You can kind of see, they still absorb photons of light, the higher, but this is going to be closer to where green yellow is. This is more blue green, and the blue cone, which most people call blue cone, is really going to absorb photons efficiently at the purple. So you can you can name those, but most people are going to call them short wavelength, medium wavelength, and long wavelength. And we already talked about the rod's absorption peak being around 500 nanometers. And that's how we perceive color, because how we combine all of these wavelengths of light in the brain via the optic nerve, it's going to be the mixture of the nerve signals representing cones from different absorption peaks. Also, this is what leads us to color blindness, because depending on how we perceive color, we can have a hereditary altercation or lack of one photoopsin or another, because those photoopsins are just going to be proteins with amino acids. If we don't have the gene for that, we can't produce that photoopsin. We can't perceive that color because that wavelength of light is just missing. And then most common is red to green color blindness, results from lack of either L or M cones or long or medium cones, causes difficult distinguishing related shades from each other, occurs in about 8% of males and about 0.5% of females. And because it's a sex linkage, it's mostly in males. And there is green, red, and yellow here. And if you are colorblind, you just don't see this shape right here. So finishing up with stereoscopic vision. Stereoscopic vision allows us to see depth perception. It's how we can judge distance to other objects. It requires both of our eyes to overlap. So when you look forward, your left eye is going to pick up a full range of vision. Your right eye is also going to pick up a full range of vision. And how they overlap and how the brain interprets that overlapping is how we can have depth perception. So what happens is it's when each eye looks at the same objects from different angles. Those angles can be judged differently in the brain, and that's how you can see how far that is away from you. Panoramic vision, having eyes on the sides of our heads. Horse, rodents are alert to predators but have no depth perception because they see a wider field, but because there's no overlap, they have no idea how far away those are. You also can have a fixation point of space in which your eyes are focused. Looking at an object within 100 feet, each eye is viewed from a slightly different angle, provides the brain with information used to judge position of the object relative to fixation point. That's why if you become a golfer, a lifeguard, a police officer doing road duty, you'll be able to determine distant objects and be able to determine how far things are away. just because you start judging this, you start seeing what things look like at distance. It says 100 feet, but if you're a golfer, you can say that's 300 yards, and you just know that's 300 yards because of how your eyes are seeing how stuff is far away. You can also judge how far something is away from one point, or if you're reading a book, this is going to be the near object. But also here, how does the eye look at do this? Does it by the medial rectus, by trying to push the eyes to look at a near object, or the lateral rectus to have it look farther away? And the pathway for this, bipolar cells of the retina are first order, retinal or ganglia cells, or retinal ganglia cells are second order. That just means these are the ones that are going to form the optic nerve. The two optic nerves combine to form the optic chiasm. The optic chiasm is that structure we talked about in the brain, or the optic cross, where the left signals go to the right, right signals go to the left. That's why they cross. Half the fibers cross over to the opposite side of the brain. That's called a hemidecussation. Hemi for half, decussation for crossing over. And the chiasm splits to form the optic tracts. Right cerebral hemisphere sees objects from the left, and the left cerebral hemisphere sees objects from the right. Each side of the brain sees what is on the side where it has motor control over limbs. So your motor control also crosses over through the corpus callosum. So the optic tracts pass laterally around the hypothalamus, with most of their axons ending in the lateral nucleus of the thalamus. So this is another sensory that goes through the thalamus for processing. Third order neurons arise here and form the optic radiation fibers and right matters of the cerebellum. Project to the primary visual cortex in the occipital lobe, where conscious visual sensation occurs. A few optic fibers project to midbrain and terminate in the superior colliculi and the pretectal nuclei. Superior colliculi controls visual reflexes and intrinsic eye muscles. Pretectal nuclei are involved in photopupillary and accommodation reflexes. All that means, look at this right here, is when they go to these other nuclei over here, this just allows them to accommodate for the amount of light coming through. So you want something set up that if you're in a bright light situation, instead of it going into the visual cortex in the occipital lobe and then kind of going all the way back, it kind of stops halfway, and then that can send signals to the muscles to look at a different area, to you can allow less light in, allow more light in, and stuff like that. So it sounds confusing, but not really. So half the fibers go through the optic chiasm, the other half just goes straight to the other side. And you can see the ones that cross, they go to different areas of the visual cortex. So this is the right eye. If it crosses, it goes to the medial side of the occipital lobe. If it doesn't cross, it goes to the back of the occipital lobe, and the same thing for the left eye. And it does this because look at how those eyes overlap. That's why you need them to cross over for you to be able to judge how far something is away, and how and that's how our scotopic vision works, and photooptic vision works, and how we can see far away and nearsighted. So some processing begins at the retina. So we can kind of adjust for contrast, brightness, motion, that type of thing. But the primary visual cortex in the occipital lobe, it's connected by the association tracts to the visual association areas in the parietal and temporal lobes, which process retinal data from occipital lobes. All this means that all the way in the back of the occipital lobes, that's called your primary visual cortex. Then you have a secondary visual cortex, which is in the parietal and temporal lobes, right in that area, that actually start to process what's happening in the occipital lobes. So here you can see, where is that object in space? Is it moving? What color is that? Have you ever seen that thing? Is that color white, gold, or black, or blue? That's where you start to interpret colors. Shape, boundaries. Is it a picture? Is it somebody's face? Do you start remembering what these are? Are these letters? Do those letters make words? So the further you go forward into the brain and into the cortex, away from the occipital lobe and forward, that's when you get higher order visual processing. And finally, anesthesia. So when we looked at this, anesthesia can be cutting a hole in the skull to let out evil spirits. That's what they had. They also had 3 things needed for effective surgery. Better knowledge of anatomy. You're like, what? After this class, you know you've already gone through chapter 1. You learned all the things about history. We no longer have to bleed somebody to let out bad blood. We don't have to cut a hole in the skull anymore to let out evil spirits. What we can do is we can look at what's causing that disease and be able to fight it more efficiently. Because we now know better anatomy, we can do surgeries more efficiently. We can actually control the infection much better by everything you've already learned in this class. We can use better anesthesia to control the pain during and after surgeries. Ether and chloroform gave way to safer anesthetics such as cyclopropane, ethylene, and nitric oxide. And all of these things mean is that we've learned all this stuff about anatomy. And what we've done is taken everything we've learned to make surgery, to make healthcare better and more effective for the patient. Hopefully you had a good class, and hopefully you're ready to take your final test in this class. Good luck.